Osmosis is the passage of water molecules across a selectively permeable membrane from a dilute solution to a more concentrated solution. When the difference in concentration between the solutions increases, the rate of osmosis increases. The rate of osmosis also increases with increasing temperature. This is a diagram to illustrate osmosis. In the diagram, we see two different regions. The region on the left is a concentrated sugar solution, which means it's a low concentration of water molecule region. And the region on the right is a water region, which is highly concentrated in water molecules. Osmosis will move water molecules, and only water molecules move during osmosis. So water molecules will move from the region that has more water molecules, and also that region is a low concentration of the sugar molecules in this particular illustration. So the water molecules will move from the right hand side to the left hand side. For osmosis to take place, there should be a semi-permeable membrane, also known as a partially permeable membrane, as we can see right in the middle of the diagram. So water molecules are going to move from right to the left until the concentration levels of both regions are almost the same. We look at some examples of osmosis in both plants and animal cells. There are salts and sugars dissolved in the cytoplasm and cell sap of a plant cell. Therefore, when a plant cell is placed in water, water passes into it by osmosis. The cell swells up as water passes into the sap in the vacuum. However, due to the thick cellulose cell wall, the cell does not burst. The cell wall may stretch, but it does not break. This makes the cell turgid. Plants are supported partly by the turgidity of their cells. When a plant cell is placed in a solution which is more concentrated than the cell's contents, water is lost from the cell by osmosis, and the cell becomes flabby or flaccid. Eventually, the cytoplasm and cell membrane shrink away from the cell wall, which collapses as the volume of the vacuum decreases. This condition is called plasmolysis. When an animal cell, for example, red blood cell, is placed in water, water enters the cell by osmosis. This causes the cell to swell up and eventually burst. In this case, there is no cell wall to prevent the outward pressure bursting the delicate membrane. When a red blood cell is placed in a solution which is stronger in concentration than that inside the cell, example, a 3% salt solution, the cell loses water by osmosis. This causes the cell to shrink and wrinkle. However, animal cells are normally surrounded by a liquid which has the same concentration as that of the cells, producing a state of equilibrium. In this state of equilibrium, even though water molecules can still move across the semi-permeable membrane, there is no net movement. This means as water from one region moves to the other, equal volumes of water from the other region will also move to the other region. In this illustration, the possible effects of osmosis on a plant cell when placed in both a hypertonic solution and a hypotonic solution are examined. To begin with, we should understand that a hypotonic solution is a solution that is of lower concentration, and a hypertonic solution is a solution that is of higher concentration. When a plant cell is placed in a hypotonic solution, the water around the plant cell will enter the plant cell. This will cause the plant cell to swell up as seen in the diagram. On the other hand, when a plant cell is placed in a hypertonic solution, water will move from the plant cell to the hypertonic solution, causing the cytoplasm of the plant cell to shrink away from the cell wall. In this other illustration, the possible effects of osmosis on an animal cell is being demonstrated. The animal cell taken into consideration is a red blood cell. When a red blood cell is placed in a hypotonic solution, in this case water to be specific, water molecules through osmosis will enter the cell. This will cause the cell to swell up. Unlike the plant cell, the animal cell has no protection in the form of the cell wall to keep it turgid. In this case, the animal cell is going to burst. On the other hand, when the animal cell is placed in a hypertonic solution, in this case a concentrated salt solution, the animal cell will lose water to the hypertonic solution and will cause the cell cytoplasm and its cell membrane to shrink, as seen in the illustration.